Hello, welcome to the next A-level video from the conscientious biologist, Ben Gallagher. This one's the fourth one in the nervous system playlist. It follows directly on from the saltatory conduction one, and we're looking at receptors now. This explains how the nerve signal is first generated so it can then be passed along the whole neuron. We're going to do a quick recap of saltatory conduction itself on the first slide, then look at some of the main receptors that you need to know about, which are the Pacinian Corpuscle and then rod and cone cells of the eye. As always, please do subscribe to my channel on YouTube if you haven't done so already. It really does help me. And remember, you can find infographics and backup information as well on both my Instagram and Facebook pages. Thank you. I'm going to go quite quickly through saltatory conduction because you should already have read the previous video. This is just to link it up to what we're talking about in the receptors. So the mechanism for saltatory conduction, if we've got a long axon here, it's got the Schwann cells wrapped around it that provide the insulation because of the layers of membrane. And remember, revise your membrane biochemistry stuff, because the reason Schwann cells can act as insulation is because they don't have the protein channels to allow the sodium and potassium ions to move through. Remember, charged particles have to move by facilitated diffusion, and if you've got membrane wrapped around the axon that doesn't have those channels, it's not going to allow that movement of ions, so it's going to insulate the electrical impulses. Now, all those nodes of Ranvier, which are the gaps, you should have a resting potential of -70. But if sodium moves in at that far node of Ranvier, that's going to change and cause an action potential by bringing in those positive charges. Some of those positive charges will then diffuse down to the next node of Ranvier, raising the resting potential from -70 to -55. Now, -55 is the threshold potential. It has to get to that threshold or an action potential won't be triggered. But as long as it hits that threshold, the sodium voltage gated channels should open at that node of Ranvier. That's going to allow an influx of sodium, which is then going to jump it to an action potential at that node of Ranvier. And then the same thing just repeats. The sodium diffuses down, raises the resting potential to threshold potential. That triggers the next action potential in that node of Ranvier, and it moves on down to the next one. What we're interested in this video is way over there. Look, where did those original sodiums come from? That's the key thing to understanding receptors, because they generate the first action potential. So let's start looking at the first one of those, which is a Pacinian corpuscle. If you remember back to the first video we did on the nervous system playlist, we looked at the overall nervous pathway and we looked at different types of receptors and the stimuli that they can receive. Now, a Pacinian corpuscle is the one associated with touch because it picks up on pressure. That pressure, of course, can be felt all over your body because Pacinian corpuscles are found in the skin. At highest density, they're found in your fingertips and your hands, because that's what you use to feel things. That's your most sensitive part of your body for touch. But you do have them distributed throughout the skin, which is why no matter where you get touched on your body, you can feel it happening. So this is the receptor for pressure or touch. Now, if I throw up here a sensory neuron again, just so you can see where we're talking about. And if at the very end, I put, just like I've shown you on previous diagrams, that series of circles there, that's representing the receptor, but it's actually a really good way of representing a Pacinian corpuscle. So if I just enlarge the end of the sensory neuron here, so you've got the very tip of the sensory neuron and the first Schwann cell a little bit closer to me. Here, just above me, would be the first node of Ranvier. Now, the Pacinian corpuscle is wrapped around the very end of the sensory neuron, so the start of the sensory neuron, and it's just layers of connective tissue with a viscous gel in between those layers. That's what's called the corpuscle, and named after the person who discovered them and described them. So they are Pacinian corpuscles. Now, what happens here to generate an action potential here to start the whole thing off is very similar to what happens in any node of Ranvier. We start with a resting potential that's established by those sodium potassium pumps exchanging 3 sodiums for every 2 potassiums to make it more positive on the outside than the inside. So the overall difference is -70. It's 70 less positive inside than outside. Now, what happens when pressure is put on that Pacinian corpuscle? It affects things called stretch mediated sodium channels. all it means with stretch mediated is when they're squashed, when pressure's put on the Pacinian corpuscle, it just stretches those channels a little bit, allowing them to open and bring sodium in to the axon at the very start of the dendron at the very start of that sensory neuron. Okay, so because their shape changes very slightly, sodium can move in. Doesn't have to be enough to start an action potential, remember. It just needs to be enough to raise the voltage inside the cytoplasm to -55, to that threshold potential. But because this is the receptor, and this is where the impulse is first generated, we don't call that the threshold potential here, we call it the generator potential. That generator potential, which is the same as the threshold potential, it's -55, is going to trigger the opening of the voltage-gated sodium channels, which of course is what's going to cause our new or first action potential in the neuron. Once we've got that first one, that's where the sodium can then diffuse down. So this is where we could have stuck our diagram from the previous page straight onto the end here to have the full start of a neuron there. So this is called the generator potential, which is at the start of the sensory neuron. So that's how we feel touch. We now need to look at how we receive light information, which is how we see. Now, the organ we use for seeing is the eye. So let's do a very, very quick run through of some of the GCSE information you should remember on the eye. I'm going to go very fast on here, take a screenshot near the end of it so you can revise it. But I don't want to spend a lot of time on this because we're talking about the receptor cells, not the organs that they fit in. But if we're building an eyeball here from a side cross section, the hard outer covering of your eye, the really tough sort of rubbery layer that's white that you can see, that's called the sclera. At the front of the sclera, you've got a layer called the cornea. It's still just the sclera, but the cornea part of the sclera is transparent. That's at the front of the eye so light can get in. The liquid that fills up the whole eyeball is called the vitreous humor. Now, lining the inside of the sclera, you've got a dark black layer called the choroid. It's black to prevent internal reflection of light. Otherwise, light would come in through the front of your eye and bounce around everywhere if it was white. By being black, it can absorb that light, stop it bouncing around to create multiple stimuli. Then you've got the front coming down from the choroid, you've got your iris. Your iris is the coloured bit that can open or close to control the amount of light that comes in. Go back to the GCSE videos if you can't remember that, because there could be questions on it on A level, even though it's technically in the GCSE syllabus. The hole, of course, between within the iris is the pupil. That's what allows the light to come through. Just to link up the diagrams here from the side cross section to the front, and if I draw lines across, you can see exactly how all these layers link up to what you see when you look at the front of the eye. So you're looking at there the white sclera that's at the top and the bottom, all the way around. The iris is then a circle, and the pupil's the hole within that. So you can put all of it together there. Extra bits, of course, or the key bit that we're talking about, sorry, is the retina. That's the photosensitive layer at the back of the eye. That's what we're going to look at in detail in a moment. That's connected to the optic nerve to carry the signals to the coordinator, which would be your brain. And of course, you've got the lens held in place by the ciliary muscles and the suspensory ligaments to pull it and focus the light. So you've got all the information you should need there about the eye. Only last label is the aqueous humor that's in front of the iris and the lens. That's the liquid at the very front bit of your eye. But that's all the information about the eyeball itself. So take a screenshot of that if you don't remember it from GCSE and revise from that. Okay. What I just want to do though is zoom in on a little part of the retina here and just enlarge it over there, because that's what we're looking at next. We need to look at the cells inside the retina, and they line up like that along the back of the retina, all kind of pointing with their pointy bits sticking forward. If you look carefully at all those cells I put there, you'll see two very distinct shapes of cells. There's ones where the back is kind of pointed and goes into a cone shape, and there's ones that are just straight like rods. That's how they're named, cone cells and rod cells. That's what we're going to look at now. So those cells that I just showed you in location, they're cone cells and rod cells, named because of their shape. The cone cells have got kind of cone shape at the back. The rod cells are straight like a rod. We're going to focus on cone cells first. Now, cone cells can detect light just like rod cells can. But what cone cells detect is the wavelength of the light that hits them. Now, pulling in a bit of GCSE physics here, you may remember that the wavelength of light gives you its colour, because light is just the visible part of the overall electromagnetic spectrum. So over the side there, I've shown you a diagram. You can see visible light there in the middle of the whole spectrum, but then enlarged into its different colours. So the biggest wavelengths of visible light are about 700 nanometers. And if you remember back to your microscopy lessons, a nanometer is 1,000th of a micrometer, which in itself is 1,000th of a millimeter. So 1 nanometer is 1,000,000th of a millimeter. So we're still talking about tiny wavelengths, but the biggest waves are red light, going down to the smallest of about 400, which are your darker blues. Okay. What your cone cells can do, they can be stimulated by those different wavelengths because they contain a chemical called iopsin. Now, iopsin is a color pigment. You have red, blue, and green iopsins. When light of the wavelength of each of those colors hits the matching iopsin, the iopsin will break down and create an electrical signal and an action potential within the cone cell. So if there's wavelengths that's red, it will hit the red iopsins. The red iopsins will create a signal which can then move down the cone cell to the end sort of furthest on that side. When it gets there, it's going to link up with something called a bipolar cell. So you've got a little synapse there, look. Now both of those together make up the receptor, the cone cell and the bipolar cell, because those are what then link up to a sensory neuron. And the sensory neuron will then carry that signal down through the optic nerve and down to the brain. So this is how cone cells work. So cone cells detect color. So remember, C, cone cells, C, color. Cone cells detect color because of this iopsin, and iopsins respond to different wavelengths of light. That's all you really need to remember for cone cells. So let's look at rod cells now. So rod cells in the same location, still at the back of the retina, but they're shaped like this, much more rod shaped. Light's going to hit them, but this time they don't detect the wavelength of light. They detect the intensity of the light. So how many waves are hitting them, how much light is hitting. And instead of iopsin, they contain something called rhodopsin. But the principle is exactly the same. The rhodopsin gets broken down when it hits light. That generates an action potential within there, links up to a bipolar cell and a sensory neuron that goes to the brain. So you can tell how bright the light is. Now, what's really, really important with rod cells is that they allow you to see really low light intensities because they can do something called summation. Okay, so I've put here one rod cell alone may not be stimulated enough to cause a new impulse in the bipolar cell. So it may have an action potential itself. Some light may have hit it, it may have stimulated. It may have caused the release of some vesicles of neurotransmitter, which could send some neurotransmitter across this little synapse between the rod cell and the bipolar cell. But if not enough neurotransmitter comes across, it won't cause a threshold potential in the bipolar cell, which means the bipolar cell won't make an action potential, won't send it on, and it'll be as if you never saw anything at all. So they do this cunning thing called summation, where you can get multiple rod cells connected to one bipolar cell. Not the best diagram I've ever done here, but you get the point. The bipolar cell can be much larger at one end, so it can feed off several rod cells. That means any one rod cell alone may not be stimulated enough to release enough neurotransmitter to cause an action potential in the bipolar cell. But if 3 or 4 or however many are stimulated enough that collectively, that the sum of their neurotransmitter moving across to the bipolar cell is enough to reach the threshold potential, then you'll get a signal along the bipolar cell and it will tell your brain, yeah, there is some light here. Now there's a problem with that. There's a bit of a trade-off, that even though the sum of the weak signals allow you to recognize that there is some light, it's very hard to tell exactly where that light was coming from, because instead of one rod cell saying there's light hitting me, here's the precise acuity of where that light's coming from, if you've got 3 or 4 or 5 and together they're saying, yeah, there's some light, you don't know exactly where it hit. Which is why, if you notice, in dim conditions, so when you're in bed tonight, turn the lights off and it goes dim, look around, everything looks a bit fuzzy. There's no real sharp edges. If you pick up a book and try to read the words, it's really fuzzy. It's actually hard to focus on them because you don't know exactly where the light is and isn't coming from, because it's spread over multiple rod cells which are summing the information together. Cone cells, on the other hand, need loads of light to see anything at all, which is why, and you can test this again tonight, turn all the lights off and you'll notice everything becomes black and white. You've probably never noticed this before if you've never been told this before, but in very dim light it's fuzzy because of summation, and it's black and white because the cone cells only work when there's quite a high intensity of light. You need lots of light to see in colour. So take a screenshot of this one, go back, take a screenshot of the cone cells if you didn't do that, but that's the key information about the 2 photoreceptor cells in the retina. Okay, we've talked lots about synapses before, we've talked about where they are and the gaps that need to be jumped. What we haven't looked at is the detailed mechanism by which synapses work. So head to Nervous System 5 next, please, on the details of synaptic transmission. If you haven't done so already, please do like this video, subscribe to my YouTube channel, and do remember to check out all my infographics on Facebook and Instagram. Thank you.